the more we learn about the perceived or purported climate benefits of no-till agriculture, I think the more we kind of raise some questions about it. And I'm really referring to the sort of more widespread herbicide-based no-till that we see out there. There had been a lot of excitement that, oh yeah, and it kind of makes sense that no-till would increase soil carbon, have climate benefits from that way. But that was a result of a lot of studies that only measured the change in soil carbon in the top 6 inches or foot of soil. So as we have more studies that dig much deeper and get down to the top 3 feet or more of soil, what we're really seeing is not much of a difference in a lot of places with no-till versus till in terms of soil carbon. What's really happening is that there's kind of a redistribution of soil carbon, meaning in a tilled system, we get a concentration of carbon at the surface, and then actually somewhat less carbon a little bit deeper. And with a tilled system, we get a little less carbon at the surface and a little more deeper.
Welcome to the Real Organic Podcast. I'm Linley Dixon, co-director of the Real Organic Project. We're a grassroots, farmer-led movement with an add-on organic food label to distinguish organic crops that are grown in healthy soils and organic livestock that is raised on well-managed pasture. You just heard from Tim Bowles, a professor at UC Berkeley's Agroecology lab, whose research lies at the intersection of agroecology, soil ecology, biogeochemistry, that's a new one for me, and plant-soil microbe interactions. Today's interview was conducted virtually after I was so impressed by Tim's presentation at EcoFarm last year on how mycorrhizae function under different management practices in tomato production. So get ready to geek out on soil life today. This is just the first part of a 2-part interview, and in the second half, we'll dive into why, believe it or not, soil health has become political and how a much more nuanced understanding of how different practices affect soil could help us redesign a food system that works better for humans and the planet. I'm here with Tim Bowles. He's the professor at UC Berkeley in agroecology. And Tim, I'm going to go ahead and let you describe everything you do at Berkeley and maybe some of your research interests as well.
Sure. Well, first of all, thanks so much for having me here, Linley. It's a real honor and pleasure to be talking with you and about the Real Organic Project. So as you mentioned, I'm a faculty member, researcher, teacher at UC Berkeley. And I run the Berkeley Agroecology Lab. And we do research on a variety of topics, everything from sort of trying to understand the rhizosphere, or this zone around the roots that is this hot spot of kind of interactions between soil organisms and plants, and how agricultural activity affects that, all the way up to really doing more interdisciplinary work about how different types of state or federal policies are affecting organic farmers in particular, and the opportunities or barriers that they might face to using different systems or practices, and then how that translates back into their soil ecosystems. So the soil is kind of the main lens that we have in our research and trying to work in organic agriculture. But we're always trying to put that in the context of this bigger system.
It's so wonderful to see this being such a sexy topic right now. I did my research in an organic research lab at the West Virginia University at the time. Yeah. There were only a couple of universities that were studying organic agriculture. And so kind of describe how the landscape has shifted. It was really rare. And I still had to take all the conventional, I had to take all the pesticide classes, and I was in the regular agronomy department, and we were just kind of the weirdos over there in the organic department. But it was around the time of the USDA creating standards, and that's why it was acceptable then to actually have some organic research. So maybe to talk a little bit about, and maybe there's still some of that animosity there towards organic agriculture. I don't know. How fringe is it now?
Yeah, that's a really good question. And I think it's going to vary a lot depending on where you are. And I feel very lucky to be here at UC Berkeley that has a really long history of both agroecology with my colleague now retired, Miguel Altieri. Um, and and even during his time and before that, we had a division of biological control here, a whole department that was focused on ecological pest management and really understanding the ecology of agricultural systems and working with farmers to develop alternatives to pesticides. That department has disappeared, and it has been part of this, I think, broader shift away from maybe understanding alternatives to our conventional agriculture. But we have this little vestige still remaining here at UC Berkeley where this, I think, this is accepted and it's valued. And certainly we have colleagues across land grant universities all over the country that are doing great work in this area. But I think you're right in that it's still, even now, I think it's still not the mainstream in terms of land grant university kind of agricultural research. But at the same time, it's gaining, I think, more acceptance in the academic world where there's this recognition, and it's coming. Why did it take so long? But it's coming that we can understand the ecology of agricultural systems just like any other ecosystem, and there's amazing life and interactions happening in these ecosystems. So from an academic perspective, right, it's becoming more acceptable to do work in these in organic. And of course, that it shouldn't have taken the sort of academic powers that be to sort of give their stamp of legitimacy on it, but it is. I do think it's something that's changing.
Yeah, it's taken a while, but it's exciting. And I loved, how many grad students do you have working in the lab? I loved meeting some of them last time I was there.
Yeah, right now I have 4 PhD students, 2 that are kind of close to finishing, 2 that are just starting out, and then I also have postdocs in my lab, which are folks who have finished their PhDs and are doing, kind of like a residency if you were, trying to become a physician, but some time after their PhD is done when they're really focusing on research.
Yeah, and what was so exciting was that they were doing research on a lot of the farmers that we know that are part of the Real Organic Project and looking at their systems and trying to give them answers about their soil and what's going on. When I first met you and wanted to interview you, it was after your eco-farm talk, just last January, and they put you in too small of a room. There were like farmers all standing in the back, and then there were some like lined out the door that couldn't get in. And I was wondering if you could just give an overview, because it was so good, what you talked about that day, because I was really moved and excited about what you were finding.
Yeah, sure.
If you can remember.
Yeah. No, I absolutely remember. I mean, these are topics that are really near and dear to my heart, but also, our central focus of our lab's work. And so this was a presentation where we were really trying to share about all of the interactions between plants and soil and microbes that affect how plants access nutrients and water, and also then how does our agricultural management either hinder or support these interactions? And so it's a good reminder that and I don't think I'm preaching to the choir here in terms of, I'm sure you're the folks listening, that soil is absolutely bursting with life, right? This is there was actually a recent study that came out that showed that 60% of the world's biodiversity exists below ground. So if we added up all of the taxa, all of the microbes, all of the plants, all of the animals, all of the world's life, 60% of those species either live exclusively in or are deeply rooted in the sense of plants, our soils. So this is more diverse than a tropical rainforest and especially when we look at the microbes that are living there, the bacteria, the fungi. the archaea. And so one of the groups of organisms that we focus on a lot in my lab, and I think for good reason, sort of have some star power, are what are called arbuscular mycorrhizal fungi, or I might, for brevity, I'll just say AMF sometimes. But these are fungi that it's a group of soil organisms, group of soil fungi that have a very intimate relationship with plants. In fact, they cannot complete their life cycle without associating with plant roots. So this is what's called an obligate biotroph. So obligate meaning like they have to, and biotroph meaning like associating with something living as part of their the way that they earn, the way that they eat. And these organisms formed a relationship with plants like 500 million years ago. And we actually think that they were what helped plants actually move from living exclusively in the water to being able to exist on land. And what they do is they help plants acquire nutrients through their wide network of hyphae that extend out into soil and get all into little soil pores inside of little aggregates. And then they actually grow inside of plant roots as well. And it's in there where this really dynamic exchange starts to happen with plant roots. The plant, it's this photosynthesis machine. It's using the energy of sunlight to create energy in the form of like carbohydrates and other types of carbon-containing compounds, and it kind of shuttles that down into its roots, helps feed these AMF. The AMF, in turn, they go out, they scavenge up nitrogen or phosphorus, and they deliver it back to the roots. So this, this is a super interesting and very mysterious in some ways group of organisms. We've really just scratched the surface in terms of our ecological understanding of what they're doing.
When you were speaking, you started talking about the streaming that kind of even goes on outside. So I had never thought of that. There's, cytoplasmic streaming like inside the fungi, but then
Right.
you talked about maybe quicker movement of water and nutrients outside the fungal hyphae. Can you dive into that? That was really fun.
Absolutely. Yeah. So this was some really recent research. There's always been a notion that, well, AMF, they might, in some contexts, they might help plants access water or help deal with low water situations. And there were lots of ideas out there about how they did this. And some of them were kind of indirect. In fact, there was some folks who thought, well, AMF aren't really moving water itself. And they're not really helping plants acquire water. They're doing other things that help plants deal with low water situations. But recently, there was a study that was a really cool experiment that was able to show that instead of water flowing within these hyphae, right? And we have to remember these hyphae are very, very small. They're just a fraction of the size of a root, even a fraction of the size of a human hair, very, very tiny. That instead of water moving on the inside of these hyphae, the water was actually moving along the surface, along the outside of the hyphae. And so as these little hyphae were getting into these little tiny soil pore spaces where there even can exist water, even when the soil overall is pretty dry, the sort of suction that's created from plant roots could almost pull this water along the surface of these hyphae. And if we think about soil, there's a lot of gaps, there's a lot of pore spaces if we're thinking down in these really tiny areas. And what the hyphae are doing is sort of helping to move water from a little tiny wet spot toward the root and crossing some of these drier pore spaces that water wouldn't be able to cross otherwise. And so it was some of the first ecological research that was showing that, yes, actually AMF do have a role in transporting water towards plants, but it's happening on the outside of the hyphae, probably not on the inside. So super fascinating work.
Because first we were just seeing that if you had crops that had these mycorrhizal fungi, that they were able to survive drought better. And so
That's
now
right.
we're trying to figure out why that
Exactly.
is.
Right.
Okay.
Right. And this is one clue to that. I think that there are some other ways, in other experiments that we've done, sometimes we use this little model system that we have where we have a tomato, it's a determinate variety of a tomato, like aroma style paste tomato. And we have, this kind of normal, the wild type version of it. And then there's a sort of a mutant version of it. This wasn't created by genetic modification or anything like that, but it was sort of created, just happened to pop up in a research context. And it has a mutation in a couple of genes that makes it mostly unable to form mycorrhizal associations. And so we can plant this wild type tomato and then this kind of mutant tomato that is in all other ways is very similar. We can actually plant them side by side. We've done this actually on organic farms in Yolo County, California, and here around in the Bay Area, and then really be able to ask what are AMF doing, right? Because we have the wild type, and then we have this very closely related genotype that doesn't form mycorrhizal associations. And what we've been able to show out here in California, where any California listeners will know, like, water is always a concern. All of our summers are always dry. We've been able to show that when there are mycorrhizae present, the plants are really changing the way that they are photosynthesizing and transpiring. So transpiration is water moving up through plants and then out in the atmosphere. It's and on our mycorrhizal plants, they were really regulating transpiration and the loss of water in a way that was better able to adapt to low water, to drier conditions relative to the non-mycorrhizal ones. So there's a lot of ways that and direct transport of water is one of them, but there's a number of different ways that mycorrhizal fungi could be helping plants deal with low water, with droughts.
Just as a side note, what is this? There's a single gene that doesn't allow mycorrhizal formation. What is that gene doing?
Yeah, it's a collection of a few. So there's this really like intimate kind of crosstalk, at the sort of chemical or molecular level that has to happen between a plant root and the fungus in order for basically the plant to kind of allow the fungus to come literally inside the root. These things aren't hanging out on the surface of the root. They're actually growing inside root cells and forming these specialized structures. That's actually how arbuscular mycorrhizae get their name, is these structures that they form inside the cells are called arbuscules, and they look like beautiful little trees almost. And so for Spanish-speaking listeners, arbol is the word for tree. It's Latin roots. So arbuscular mycorrhizal fungi, they're named for that, for these super intimate structures that they form. So they're
maximizing the surface area in there, so that we can do all that exchange, right?
Exactly. All the surface area in there, so they can get energy from the plant, exchange it with nutrients. And so you can imagine
plants and fungi both have cell walls, right? So, and those are gone too. So we've got membrane to membrane, tight, close interactions.
Yeah, exactly. And so you can imagine, like, this is a relationship that has formed over millions and millions of years of evolution. And so there's a set of genes that are involved in this kind of molecular level communication between the plant and the fungus, in order for the plant to say, okay, yeah, come on inside. It's okay. I recognize you. And those are the genes that have been disrupted in this mutant. So just to be clear, this is not something that we are using for For cultivation, this is purely just like a research model system to help us understand what AMF are doing.
And so do we need to be talking about, do you pronounce it glomalin or glomalin when we're talking about kind of all of that stuff that's streaming on the outside? Is that part of your research too?
We haven't looked too much at glomalin, but it is, it's really important constituent of soil that we think of as helping to glue a lot of soil particles together and help form soil aggregates and soil structure. It's not something that we've looked at too much in my lab, but definitely in a really important product of all of this microbial activity that helps form soil structure.
When I was mostly taught about this, it was phosphorus. That was the big nutrient that was limiting, that kind of allowed the plant to say, okay, I need you because I'm phosphorus deficient. Are there other nutrients that are at play here? I've kind of heard recently some talk of nitrogen. What else is going on?
Yeah, great question. Yeah, historically, phosphorus was the big focus, and this was for good reason. In that, if we think of roots being able to access nutrients in soil, there's a couple of factors here we need to think about. One, what are all the spaces that roots can get to or not? And then how mobile are nutrients in the soil, right? And the forms of phosphorus that plants can access in soil are typically not very mobile. They don't move very far. And so plants' access to them is really limited by where the plant roots can go. And so it kind of makes sense that evolutionarily, the main sort of nutrient that plants and AMF are trading is phosphorus, because the hyphae can explore a lot more of the soil than the roots can and help get them access to these more immobile nutrients. Now, nitrogen, on the other hand, there's two main forms of nitrogen that plants can access in soil. There's some exceptions, but they mainly are looking for ammonium and nitrate. Nitrate is very mobile in soil. And I want to say this is even in organic systems, right? Where organic amendments are being added, where you're relying on legumes. All of this organic material, right, is being processed in the soil and turned into ammonium and nitrate, even if those aren't the fertilizers that you're adding. So nitrate is a little bit more mobile. So there was some thinking that, AMF maybe aren't helping too much with this because, it's mobile, it's gonna flow towards plant roots. But what we've seen in a number of studies is that we know that AMF can take up forms of nitrogen, particularly ammonium. They can transport it through their hyphae. There are sort of these, molecular transporters, these enzymes in the roots that accept ammonium from AMF. So we know that the machinery is there. And in some of our research using this tomato model system, we've shown higher concentrations of nitrogen in the mycorrhizal genotype versus the non-mycorrhizal, and this is in real field settings. This isn't in a greenhouse. And so we do think that they're playing a role in nitrogen nutrition as well, alongside the water and the phosphorus. The last thing I'll mention here is that I think it's important for all of us to remember that if we think evolutionarily, over a long time horizon, and association with AMF is the norm for plants. It's not the exception. About 80%, 70 to 80% of land plant species form associations with AMF. So this is really the norm. So when we're thinking about all the things that they do, we have to remember that this is part of this bigger picture of what a plant is, right? This idea of the holobiont. I don't know if that's a term that's familiar to anybody, but it's this idea that we can't think of ourselves as just an individual organism, because our baseline, even as humans, thinking about our gut microbes, like our baseline actually is association with all of these other organisms. And the same is kind of true for plants and AMF. This is, with some exceptions, this is kind of their normal situation.
Yeah, I love that idea of just, you can't think of yourself, alone without
Right.
all the life that's around it. There's so many places I want to go. I do want to kind of figure out, the organic farmers have so many different practices. And so I know you're kind of trying to take a look at which practices are affecting mycorrhizae, and you presented some of those findings that day. So maybe kind of set us up for what you found back then, any new research you have, and questions that you still have.
Sure. Yeah. So I want to think a little bit about, because I always like to share a bit about the ecology of these organisms so that it helps us understand how different practices might affect their abundance, their diversity. So how many there are, how many different ones there are. Yeah. And so, for instance, one of the practices that we've looked at is cover cropping. In an annual cropping system, in the absence of cover crops, there's typically can be fairly long periods where there aren't many living roots in the ground. And so, as I mentioned at the beginning, AMF require plants to complete their life cycle. They're not like other fungi that can go and decompose organic matter on their own. They actually don't do that at all. Other AMF are getting all of their energy from living plant roots. So you can imagine if you have a long time period with no living roots in the ground, a lot of the living AMF are going to disappear. What's going to be left are their spores. We can think of spores as one of the ways that AMF propagate themselves. It's, sort of like their seeds. You can think of it like that. And so what's going to be left are those spores. And so when something new is planted, those spores are going to have to germinate. They're going to have to grow towards the roots. They're not going to form those associations as quickly. And we're also going to be kind of selecting for types of AMF that can survive these longer periods without plant roots. So it might also affect which AMF taxa, which AMF species are there, and potentially their diversity as well. So that's something. So in our work on cover cropping, we've done what's called a meta-analysis, which is where we look at any study anywhere in the world that has looked at cover cropping and their effect on the abundance of AMF. And what we see is that there's about a 30% increase on average across all these different places in the abundance of AMF with cover cropping. So that's one practice we've looked at. Another PhD student that was in my lab, Aide Guzman, who's done just amazing work here with farmers in California who do a lot of polyculture, a lot of intercropping. She was interested in how polyculture intercropping affects the diversity of the mycorrhizae that are present, and found that about twice as many AMF taxa are present, or AMF species are present, when there's intercropping versus where there's more monocropping. And
that's how many is the baseline there? Twice as many from what?
Yeah, it's in the kind of low dozens. Uh-huh. So I mean, interestingly enough, like AMF globally have a few hundred different taxa, different species. They're actually not relative to other groups of microbes, they're not a super diverse group, but we know that they do different things. We know that they have different traits, they have different functions. And so just like, there's this general idea in ecology that, well, if you have more diversity around, there's going to be more of a likelihood that those organisms will play different roles or will be adapted to different conditions. And so what we sort of hypothesize here is that more diversity of AMF might mean that those AMF will be able to sort of do a more complete set of functions. Maybe some of them specialize more in phosphorus transport. Others maybe are better at nitrogen, or some of them like to associate with tomatoes more than they do with grains, right? So think of all these different ways where you might have slight differences, and a more diverse community of AMF means that you might be more likely to have a more complete set of functions, especially as conditions change.
And in general, these species aren't very host specific. They have many hosts that they can infect.
That's right. Yeah, they're not particularly host specific. That's a really good question. And we can think about this like kind of generally. There are like hundreds of thousands of different plant species. There are just in the hundreds of different AMF species, but most plants form associations with AMF. So even just based on that, we can say, oh yeah, AMF are kind of generalists. They're not too picky usually. Maybe some, but in general, not too picky about which plants they're forming associations with.
And in agricultural systems too, is it safe to say that the type of mycorrhizae are these AMF, and in more forested systems, you've got different kinds of mycorrhizae that might be there? Maybe kind of describe the difference, or maybe it's not even forested, but more perennialized systems.
Yeah. So there's two other groups of mycorrhizal fungi. They're pretty different. One are called ectomycorrhizal fungi. These are the ones that you find more in forest ecosystems, and they're pretty different. They don't actually make it inside the roots of the plants that they're associated with. They're kind of hanging out on the outside, so that's why they're ecto. AMF are sometimes called endomycorrhizae. Ectomycorrhizae are pretty different. They can decompose organic matter. They don't have to associate with plant roots. They're doing some of the same things in terms of delivering nutrients in exchange for energy, but their life history or their ecology is a little more flexible. But even in agricultural systems, a lot of our cultivated trees are also AMF dependent, like the prunus genus, which, stone fruit and almonds, and those are AMF dependent. So even in more perennialized systems in agriculture, I mean, unless you're really talking about agroforestry systems that have a lot of our cultivated, domesticated species are still AMF dependent.
One of the things I learned was that, and I got to play around with them a little bit, if there was too much fertility in the pot, we actually didn't get those interactions happening. The mycorrhizae were not accepted by the tomato plant. Is that, would you say that fertilizers affect whether or not, these interactions happen? And is there a difference between maybe an organic fertilizer that's not kind of readily available versus a synthetic fertilizer? How is that affecting this?
Yeah, that's such a good question. And we've done a bit of research on this. In general, we think of AMF and plants as having a mutualism, as being like one of the best examples of a mutualism, right? A mutualism is where, both parties in the interaction benefit, right? Plants get their nutrients, AMF get their energy, the world is good. But sometimes that's not always the case, right? Mutualism is not always a mutualism. It can be somewhat context dependent. And one of those important contexts is how much available nutrients are there. So you could imagine from a plant's point of view, if they're in a situation in a soil where there's a whole lot of available nutrients, a whole lot of available phosphorus, a whole lot of available nitrogen, well, they can get a lot of the nutrients they need just from their roots. So why spend all this energy on the mycorrhizae? And so plants have some control, and the fungus have some control, but plants can sort of like down-regulate or sort of prevent some of the associations from happening in really well-fertilized contexts. And that can ultimately affect the extent to which plants receive benefits, or we get an increase in, say, productivity with AMF. Phosphorus is probably the most studied and example of this, where, and we actually did an experiment at a long-term research trial that was happening at UC Davis. And this research trial had been going on for about, almost 25, 26 years when we were working on it. And it's a trial that compares an organic tomato corn rotation, which is relatively common in that part of the Sacramento Valley. It has cover crops. It's a tilled system with a conventional tomato corn rotation, a lower synthetic input, but still some synthetic inputs, tomato corn rotation that included cover crops, and then an extended kind of tomato corn alfalfa rotation. And we planted our tomato model system, our mycorrhizal and our non-mycorrhizal genotypes across each of these farming systems to say, okay, well, what's the effect of this like 25-year history on the way that AMF were functioning with plants?
Was
what we found really
no-till?
The conventional, they were all tilled.
Till.
Yeah.
Okay.
Yeah.
So there wasn't like a no-till with.
There wasn't a no-till component to this. It was really about sources, organic or not, so like sources of fertility and pesticides, and then with or without cover crop, and then this extended rotation with 3 years of alfalfa.
Uh-huh.
And we were really surprised. We went into this thinking, obviously, the organic system is going to have the most mycorrhizae. They're going to play the biggest role in helping plants produce biomass and tomato yield. And we were really surprised to see that there was almost no difference in growth between the mycorrhizal and the non-mycorrhizal genotype in the organic system, suggesting that AMF weren't really playing much of a role. They weren't hindering plant growth, but they also weren't helping very much. We were looking at yields as kind of just the ultimate outcome here. Where we saw the most benefit was in this rotation, and also the highest tomato yields was in this corn, tomato, and extended alfalfa rotation. And so we were trying, and sort of intermediate benefits in the conventional and the lower input. So we were really scratching our heads about this, like, what is going on? And we started looking at levels of available phosphorus in soil. And that's where we started to get some clues. The organic system had, by a pretty wide margin, the highest levels of phosphorus. We were using a measure of available phosphorus called Olsen phosphorus, which is, other states use melic, or there's different extractions that you can do to measure available phosphorus. And this really came from a history of using a lot of poultry manure in the organic system and basing the rate of poultry manure additions on plant nitrogen needs. And so poultry manure has a relatively low ratio of nitrogen to phosphorus. So what was happening was, with the history of poultry manure applications, there was kind of more phosphorus being added to the organic system than was actually needed. So levels of soil phosphorus were building up. Mm-hmm. And so what we ultimately think happened is, like, this high level of phosphorus fertility was causing plants to say, you know what, like, I don't really need the mycorrhizae as much.
Yeah.
And so it's a good just reminder around just unintended consequences of our management, right? And of course, we're always, folks are making decisions for a whole variety of reasons. But it's just a good reminder about potential unintended consequences and the complexity of some of this ecology that's happening.
Oh, okay, so many questions about that one. We're running out of time. We might have to do a two-part interview.
Sure.
Here.
It's been a really fun conversation.
I went to Washington, D.C., and, I was lobbying for organic to be more supported in the Farm Bill. And across the board, it was like, organic is not climate smart. You guys till. And my experience across the country is that the best organic farmers were actually growing these huge cover crops I'm
Right.
thinking of, I mean, in California, there's, Full Belly and I
Yeah,
know they maybe were part of some of these experiments, Phil Foster and
that's right. Yeah, that
that
Jeff
part.
Mitchell, yeah,
Yeah,
was
just to kind of
working with.
reduce
Yeah,
tillage. They were
right,
playing around with no-till, but it was really difficult for them. But even across the country, in the Midwest where it rains more, Mark Askegard produces grain, and
right.
he's growing all his own fertility. He doesn't use any
Yeah,
off-farming.
that's amazing.
He's doing
Yeah,
that with cover crops, and same thing with this woman Emily Oakley, who's been to Eco Farm, Three Springs Farm. So for me, these were the best farmers are growing their own fertility in the form of cover crops. They're
right,
flail mowing, maybe they're somehow incorporating, often quite deeply.
right.
Um, to me, I was like, this is the best kind of farming there is.
Yeah,
But we have this real difference of opinion. I think there's a lot of people with really good intentions that are saying that that kind of farming is not good,
right.
because you till and you're destroying mycorrhizae. And I'm just, curious, even if it's once a year and you're incorporating that cover crop,
Yep.
I'm thinking those spores, those mycorrhizal spores are surviving that.
They are
Is that
surviving that.
different from like a
Right,
tillage or an herbicide burn down, right?
right.
Maybe even more beneficial because the mycorrhizae, do we even know how they're affected by herbicides? We certainly know
Uh,
from fertilizers it's not great.
Yeah, and I think well, just to get to start with your first question, yeah, the spores are surviving tillage, right? Your mycorrhizae are not going to disappear from a tilled system. In that same global meta-analysis that I mentioned, where I talked about cover cropping, we also looked at reduced tillage and no-till, and you do see a similar increase in abundance that you do with cover cropping when you move to reduced or no-till, about a 30% increase in abundance of AMF. But at the same time, in the cover crop systems that had tillage, you also saw the benefits of cover cropping. So my point is there are kind of multiple ways of getting at similar kind of outcomes. From a strictly mycorrhizal standpoint, the combination of roots in the ground and minimal disturbance is probably the best. But at the same time, the predominant forms of no-till that we have in this country are all herbicide and transgenic based, with herbicide with resistance to Roundup. So I think of those no-till systems as being kind of a different category, in the sense of like we need to look at the trade-offs of that, and not get too hung up on and for organic farmers not to get too hung up on the soil disturbance that they do have to do. I am not worried about mycorrhizae disappearing from those farms. And for instance, going on long-winded here, but the farms that Dr. Ide Guzman was also working on comparing polyculture and monoculture vegetable systems. Those are all till-based systems. So even in the background of tillage, we saw this big increase in diversity with the AMF. And so it's not as if tillage is really the wiping these organisms out. That I do not think that's the case. And the last thing I'll add is maybe getting a little bit on a soapbox here is I think the more we learn about the perceived or purported climate benefits of no-till agriculture, I think the more we kind of raise some questions about it. And I'm really referring to the sort of more widespread herbicide-based no-till that we see out there. There had been a lot of excitement that, oh yeah, and it kind of makes sense that no-till would increase soil carbon, have climate benefits from that way. But that was a result of a lot of studies that only measured the change in soil carbon in the top 6 inches or foot of soil. So as we have more studies that dig much deeper and get down to the top 3 feet or more of soil, what we're really seeing is not much of a difference in a lot of places with no-till versus till in terms of soil carbon. What's really happening is that there's kind of a redistribution of soil carbon, meaning in a tilled system, we get a concentration of carbon at the surface, and then actually somewhat less carbon a little bit deeper. And with a tilled system, we get little less carbon at the surface and a little more deeper. So the story around climate benefits and kind of herbicide-based no-till is I'm not super convinced about, and I think others are also raising some questions about that.
Well, we've got our work cut out for us because they've done a good job in Washington, D.C., convincing everybody organic farmers are the problem. Are you open to another part 2? Because I still have more questions.
Yeah, absolutely. Let's do it again. This has been a really fun
good. Okay. Yeah, no, I'll honor your time, and we'll figure out another time to continue the story. Thank you so much, Tim.
Thanks, Linley. Okay.
Bye-bye. Thank you for listening to the Real Organic Podcast. Be sure to join us next time for the second half of this important interview with Tim Bowles. Our movement is growing because you are subscribing and sharing these podcasts with your friends. So keep it up and leave us a rating and a review as well. You can find a video version of this interview on our newly designed website, realorganicproject.org, or on our YouTube channel. And you can join us every Tuesday for a new episode featuring voices from the organic movement.